Hello, my name is Jeffrey, and I am a PhD candidate in Dr. Megan McCain's Laboratory for Living Systems Engineering. Today, we are going to fabricate 5% and 10% weight per volume gelatin hydrogels that are cross-linked with 4% microbial transglutaminase. Here's the container of gelatin that we will be using today. This gelatin is similar to what might be found in Jell-O or marshmallows. This particular brand was isolated from pigskin. We'll begin by adding 20 milliliters of sterile filtered water into 3 tubes. Notice that water on its own has a low viscosity. So next, we will measure 4 grams of gelatin. And we'll add this into the first centrifuge tube in order to create a 20% gelatin solution, or 4 grams and 20 milliliters. To next create a 10% solution, we will add 2 grams of gelatin into 20 milliliters of water. And you can see here that gelatin powder doesn't dissolve completely and is really viscous at room temperature. And so it needs to be kept in 65 degrees water bath for an hour. And I'll vortex the solutions briefly to encourage the gelatin to dissolve as well. So now once the gelatin solution is dissolved, it will remain a viscous liquid at 37 Celsius. But we want it to be a solid at 37 since this is the temperature of the incubator in which we culture cells. Thus we need to add a cross-linking agent that will make this a solid hydrogel at 37 Celsius. The cross-linking agent that we have chosen is called microbial transglutaminase, or MTG. Interestingly, MTG is actually heavily used in the food processing industry with the colloquial name meat glue, and is often used to improve the texture of meats such as chicken nuggets. So we'll be preparing an 8% weight per volume solution of MTG in this third tube. To do so, we'll measure out 1.6 grams of MTG and add 20 milliliters of water into this third tube. And I'll again vortex this mix before adding it to the water bath to ensure it dissolves completely. And finally, to create the final hydrogel, we will be mixing equal parts of gelatin and MTG to initiate the cross-linking of the hydrogel. This step will also dilute the concentrations of the gelatin and MTG by half, leaving us with 2 different mixtures, a 10% gelatin 4% MTG solution, as well as a 5% gelatin and 4% MTG solution. We'll mix these parts together using a planetary centrifuge. A planetary centrifuge works using both revolution around the center, similar to a regular centrifuge, and rotation of the individual sample around its own axis to cause material flow. This is called a planetary centrifuge because planets in space similarly undergo revolution about the sun and rotation around their axes. And here I'll have to adjust the balance weight to ensure the centrifuge spins properly. Otherwise the machine will shake due to imbalance and fail to homogeneously mix. The solution we can take out of the centrifuge now, and we'll leave it out overnight after pouring it into this petri dish. And this will allow cross-linking to finalize, and it will result in a hydrogel that is stable and solid at 37 degrees Celsius. Cells can now be grown directly on this hydrogel surface, since gelatin, a derivative of the extracellular matrix protein collagen, is naturally adhesive to the cells. So looking here, varying the concentration of the gelatin will change how much collagen there is to cross-link, altering the elastic modulus and color or shade of the hydrogel. As you can see, the 10% hydrogel will deform less under the same pressure than the 5% hydrogel, and to demonstrate this, I'll use my finger to press down on the hydrogel.
Hey everyone, so next we will be fabricating polydimethylsiloxane or PDMS substrates. As mentioned in class, PDMS is an inert, transparent, mechanically tunable, and gas-permeable elastomer, and for these reasons it is a commonly used biomaterial. To make PDMS, we will need to mix the base and cross-linking agent. The standard recipe for PDMS is 10 parts base, to 1 part of the cross-linking agent. So we will go ahead and measure out 10 grams of the base and add 1 gram of the cross-linker in order to obtain a 10-to-1 mass ratio. PDMS is very viscous, so in order to mix these components well, similar to the gelatin demonstration, we will need to use a planetary centrifuge to mix for 1 minute. As you can see, mixing in the planetary centrifuge introduced a lot of bubbles into our solution. To remove these bubbles, we will need to degas the solution using the planetary centrifuge for another minute. So we'll set this to memory 3, which will mix for 0 seconds and degas for 1 minute. During this process, the shear stress caused by rotation will cause bubbles at the surface to be destroyed, and reduced pressure inside the centrifuge will cause larger bubbles to expand and be destroyed. And as you can see, this successfully removed bubbles from our viscous solution. So PDMS is a thermosetting polymer, so now that we have mixed and degassed the prepolymer solution, we can add the solution to a petri dish and cure at 65 degrees Celsius for 4 hours in order to obtain a solid PDMS substrate. So changing the ratio of base to crosslinker can alter the stiffness of the substrate. We've prepared a 5-to-1 mass ratio, which we've also added food dye to make it more visible. In addition, we've also prepared a 25-to-1 mass ratio, which received 5 times less cross-linking agent for every 1 part base. And as you can see, this is a lot softer and can be deformed more easily, whereas the 5-to-1 ratio doesn't deform as visibly in response to physical touch. In addition to using PDMS to modulate stiffness of substrates, we can also use it to replicate features of a silicon wafer. This can result in PDMS stamps for micro contact printing or micromolding, and can also be used to replicate channels for microfluidic devices.
Hello, I'm Dr. Rexis Hall, and I'm a postdoc in Dr. McCain's laboratory. I'll also be a guest lecturer this semester for the lecture on bioreactors and oxygen control. Here we have a silicon wafer that we have previously cured PDMS on top of to make some PDMS pieces where we replicated the features of microfluidic devices. So here you see these 2 channels in this particular design. And we're going to want to bond that to close the channels up against a glass cover slip. So to do that permanently, you're going to need plasma. So here's the plasma chamber, and I'll place both the PDMS piece and the glass slide inside. And there they are. We'll shut the door. And first we're going to turn on the pump. And once the pump is on, you have begun to create a vacuum, and so the door is sufficiently closed. And we want to wait to get pretty negative pressure before we make plasma, and you can look at these bubbles in the oil on the outside, and once you have like a cone of bubbles, you can hit power. And once power is on, we will turn on the plasma, and you should see it here. Like the purple in this chamber means we've created plasma. We plasmid for 90 seconds, and now I'm doing things in reverse, shutting off the power, shutting off the pump, bleeding in air to come back to atmospheric. And here I can take out both the PDMS and the cover slip, and I'll flip that design of the channels onto the cover slip, line it up, and apply pressure using the back of these forceps to make good contact. And now I have an irreversible bond. What do you know? So using the same kind of method, I've made this Y-channel microfluidic device, and I'm going to use some food coloring in water to perfuse through the channel. So here I have tubing that I am putting in the inlet using these tweezers that has green dye, and similarly in the other inlet I'll be putting in fluid that has yellow dye, and I'll perfuse this Y channel. And what you can see here is laminar flow through that channel, so very little mixing and parallel flows of both dyes. But this channel, this one has a herringbone design, and the reverse staggered herringbone ridges allow the channel walls to introduce transverse flow patterns and enhance the mixing. So that's what you've seen. So altogether, I'm showing you these microfluidic devices that you can fabricate from PDMS, and based on the channel design, you can change things like flow and, in this case, enhance mixing.
So similar to the last lab demonstration in which we had taken fluorescence images of aligned C2C12 cells, we have prepared PDMS stamps with lines that are approximately the width of cells, which will be used to micro-contact print cell adhesion proteins in order to promote cell alignment in cardiac myocytes, or heart muscle cells. Although we can't see these lines with the naked eye, under the bright field microscope, it is clear that this stamp has microscale features. These PDMS stamps were molded on a silicon wafer that had this exact line pattern. We next used these stamps to transfer a cell adhesion protein called fibronectin onto cover slips, and cardiac myocytes were then grown on these substrates to promote cell alignment. So next we immunostained aligned cardiac myocytes so that we can look at sarcomeric alpha-actinin, which we also saw in C2C12 cells and makes up sarcomeres, or the basic contractile unit in muscle cells. And we also stained for DAPI, which stains cell nuclei. So we'll select the appropriate filters, add a scale bar, and capture the image. And as you can see, cells are mostly restricted to the regions where we had micro-contact printed fibronectin. These dark regions, which are devoid of any cells, were blocked using Pluronic F127. And just as a comparison, we've also immunostained isotropic tissues, or tissues that do not have any particular directionality. And as you can see in the alpha-actinin stain, these cells and their sarcomeres are not aligned with one another. And we can see the same thing in the cell nuclei stain, where cells are sort of everywhere rather than in desired locations. And we'll go ahead and capture this image as well.